worthy of a note in evolution, but somebody came to my lab and said, why don't you try putting it in Nature? I said, no way. Nobody knows me. I'm an assistant professor. But sure enough, they published it. And it was designed to explain why this phenomenon of heterogenic sterility actually occurs. And there's two explanations. And most of these, the theory and explanations have actually been worked out by Alan. The first one is simply that males, or the heterogenic sex, is preferentially affected simply because the alleles causing sterility are recessive. Oddly enough, this is called the dominance theory. It should be called the recessivity theory. If genes causing sterility are recessive in hybrids, because they interact with the hybrid background, then the heterogenic sex will be more sterile or more inviolable because every gene on that X chromosome is expressed, whether or not it's recessive or dominant. Whereas in females, any recessive gene that causes sterility on the X will be masked by a gene on the X chromosome from the other species. So that's called the dominance theory. There's been a lot of mathematical work done. And the other explanation, the main one is, well, you can't see it, it's called the faster male theory, that males are preferentially sterile and viable simply because they evolve faster than females. Maybe because of sexual selection, although that's not necessarily the case with sexual selection. And if the byproduct of fast evolution due to sexual selection is sterility or inviolability in hybrids, then the males will be preferentially screwed. That doesn't work too well as a theory because in other groups, like birds and butterflies, it's that the heterogenic sex is female. And so sexual selection still takes place in the males, but it's the females that operate. So the dominance theory is always the one that seems the most widespread and intuitive to me. It's a widely expressed theory, widely believed. Remember, Haldane's rule is one of the two great rules in evolutionary biology that has almost no exceptions. The other one is, if one sex is brightly colored or has displays because it's the males. Okay, those are our two laws that we have in biology. This is one of them. So explaining it becomes somewhat of an interesting endeavor. And the dominance theory makes a prediction, which is that if you put an X chromosome into a male, as opposed to any of the other chromosomes, that's going to make it more sterile, because that X chromosome will express every recessive gene that causes incompatibilities. So here is a backcross to, I believe, Drosophila cucuba, a QTL map showing where the genes are located that cause sterility in the male hybrid. And you can see that with these exceptions of two blips, every single one of them is on the X chromosome, and they're whopping big effects. So this data, which we replicated in other species as well, is pretty strong evidence for the fact that, yes, recessivity of genes that cause hybrid problems is a pervasive phenomenon, not just in this group of species. This result you see over and over again, not only in other species, Drosophila, but in other species of animals, and even plants sometimes. Okay. So that takes care of, Trudy McKay, in collaboration with our lab, is actually doing introgression analysis. We want to actually isolate these sterility genes. It'll probably take a long time, but we've done some introgression analyses, and hopefully in 2 or 3 years we'll have some data on that. The last sort of QTL analysis I'm going to turn to is the genetic basis of sexual isolation of these species. Because sexual isolation, we think, is really a pretty important mechanism of speciation. In species that are sympatric, that live in the same area, it seems to be the genetic barrier that evolves faster than almost anything else. Okay. So it's interesting to figure out, well, what are the genes involved? Are they the same genes in males and females? Where are they? How many are they? And what is their normal function? We don't really know any of the answers to that question except where they are in this state, but we're working on it right now. In fact, there's not a single gene in animals that's been identified and published as such that is known to cause sexual isolation. So this is like a wide open field for people doing genetic analysis, especially of behavior. So these species are strongly sexually isolated. You can see this from these no-choice studies where you simply confine a single male and a single female in a glass vial, and you leave them there for an hour, and you have somebody watch them and see if they mate or not, the two conspecific matings and the two heterospecific matings. You can see that after an hour, most of the conspecifics mate with each other pretty well, 60 to 70%. The heterospecific matings don't do too well. This shows the sexual isolation under no-choice conditions. And in fact, you can see that this particular mating between Drosophila santhanae females and Drosophila cucuba males is very, very hard to get. And it's hard to get because although these cucuba males court the gangbusters of these females, the females just kick them off. They don't like them. So what's really changed here is something in the female preference for these males. The males still court these very avidly. So we have two traits to genetically analyze here. The female preference, which has changed between these two species for Drosophila cucuba males, plus the trait, which is difference between the males that allows Drosophila santhanae to recognize its own males but not to recognize the other species as an acceptable mate. Okay? And so you can do PTM analysis of this. You simply do the genetic constitution of each fly using molecular markers, and then you see whether that fly is accepted by a male or a female of the right species. And you can imagine that's a tremendous amount of work. Anybody that works on animal behavior knows that things are messy, depends on the barometric pressure, whether you're playing REM in the laboratory rather than the carpenters. It's a huge difference. So you have to do a big sample size in order to get any kind of reliable results. So that's a slow on Alan, by the way, who listen to REM.
I think the slur goes the other way.
So there's some rudimentary PTM analysis that we're just in the first stages of. We intend, perhaps someday, to isolate the loci responsible for differences in female preference and male traits, but it's a hard slog to do that. And anyway, this is the PTM analysis showing where the regions are in Drosophila xanthomea females that make them not like Drosophila cuba males. They're all in the third chromosome, pretty much. Okay, we don't know how many genes are in here, but there's a huge effect of that chromosome. Maybe there's only one or two loci involved. We have no idea, but we're doing introgression analysis now to try to find this out. And these are the genes. This shows the mess that you get. And these are the genes in Drosophila cuba males that make them unacceptable to Drosophila xanthomea females as mate. You can see there's also in the third chromosome, but the effect is not nearly as significant. But they're in different places. This just confirms what we already know, that the genes for male trait are different from the genes for female preference. It could hardly be otherwise. Okay. Now, after we did these sexual isolation genes, I had another idea. It seems like a stupid idea. This is the kind of idea you can only work on after you have tenure, preferably after you're a full professor, because you know it's likely to fail. We all have these kinds of experiments that we always really want to do, but we can't do until we have our NIH grant already in hand. I wanted to see if sexual isolation of these flies could be changed. Because after all, these flies meet each other in nature and discriminate against each other in nature, not in the glass tubes full of food in the laboratory. So, and you'll see at the end of this talk why I wanted to do this. There really is a reason, folks. So what we did was change the environment of the flies to see if that could change the degree of sexual isolation. And these are some of the things we did. Instead of using glass tubes, we used these huge cages. We didn't have the cat in there. You just got it in there. That's our mascot for one summer. And lo and behold, what we found is we could do a lot of different things to these flies. We could crowd them, we could starve them, we could change the proportions of the two species. We could even make little mock jungles. This is really stupid experiment. With begonias and dirt and dried figs, which we thought, well, maybe this is what it's like for flies. And then we let the flies mate, and we never found any effect on sexual isolation. It was always the same strength and degree, no matter how we measured it. Only one factor could be altered that would actually change the degree to which these animals discriminate against each other, and that's whether or not they can choose each other as mates. If you have choice, if you're a Drosophila xanthomea and you have two males to choose from, you will more often choose the right one than when you have no choice. Okay? So sexual isolation can be strengthened if you allow the flies a choice, but you can't change it a lot. Okay? I call this the jail effect, the effect of the lack of choice and how but you cannot change the direction of sexual isolation. You can make it a little bit stronger by giving them choice, but you can never make that rare mating between male females and male females happen with any appreciable frequency at all. Okay? That, again, you're going to have to wait till the end to see why we did this experiment. But believe me, it's not just some stupid idea I got in the middle of the night. I mean, maybe stupid, but it does tell something. Okay, so finally, what's the advantage of working with two species that actually have a hybrid center? Well, there are several questions you can answer about speciation that you cannot answer between the species that live in different places. First of all, is there the phenomenon of reinforcement, which I'll mention in a minute. How much introgression is there between these species in nature? In these meetings already, you've heard a lot about islands of speciation, where species are different, maybe only in a few places are the same everywhere else. You're going to hear a lot more about that in Ottawa. And finally, what factors actually prevent these species from introgression? Where they live together in nature? So briefly, as I sort of wind down, I want to talk about these three areas, and I hope we'll get through in time. Reinforcement, and if you study speciation, you'll know about reinforcement. It was an idea that was much beloved with Theodosius Dubchansky, my academic grandfather. It's a phenomenon, an evolutionary phenomenon, that is supposed to occur when two species have overlapping ranges. Okay, so there's an area of overlap and an area of outlier. And Imagine, for example, that B is Drosophila ecuba, so this non-intersecting area represents not only the African mainland, but the area of Santa Tomé at low altitudes, where they do not encounter the other species. Here's the hybrid zone between 1,100 and 1,400 meters, and there's Drosophila Santa Tomé up at the top of the mountain. So, in the area where both species live, there's a selective pressure that they don't encounter anywhere else, and that's the pressure of hybridization. If you meet the wrong species and mate with it in an area of sympatry, you lose a lot of reproductive investment because half of your offspring are sterile, the males. So wherever there is this disadvantage of hybridization, usually occurring after zygotes are formed, there will be natural selection to increase the degree of mate discrimination between the species. And that's called reinforcement. So if you see a pattern like this, what you expect natural selection to do is actually increase the mate discrimination in this area here compared to species areas where they're allopatric. So all you have to do is test the level of mate discrimination between strains taken from the same area and strains taken from different area. If reinforcement occurs, you expect it to be higher from the hybrid set than in other areas. Okay. One of the most well-known recent experiments of this was done by Muhammad in Drosophilus persimilis and Drosophilus here. He found it, reinforcement. We did not. Despite the fact that they do hybridize, crucially in this hybrid zone, and this is a busy slide, don't bother. This is Drosophila ecuba females taken from the areas of allopatry and areas of sympatry, and we measured sexual isolation with respect to the other species, expecting that it would be higher from the areas of sympatry. In fact, what we got was it's exactly the same. You can't get much closer than that. There is no evidence for reinforcement having involved in the species. And that's very odd, because if we do this experiment in the lab, we put the species together for, say, 5 to 10 generations, we get this mate discrimination evolving very quickly. Okay. But we don't get it in nature. But we do get a form of reinforcement in nature. Reinforcement for something different from sexual isolation, post-mating pre-zygotic isolation. What it means is that if you are, and this only works in Drosophila ecuba, if you're a Drosophila ecuba who lives in sympatry in the hybrid zone with the other species, as compared to those strains which don't, and you make them with the other species, the ones that live in sympatry do not have anywhere from 20% to 50% as many offspring. They simply, we're not sure what this reflects, but they simply dump their sperm, which we think is what's happening. When they mate the wrong way, they get rid of the sperm. This is our anthropomorphic description of what happens. But we know that this is actually a selective advantage to do that, because the flies in sympatry that dump this foreign sperm come back into mating condition a lot faster, and will mate with the right species a lot faster than these species, which are still pumping out those heterospecific eggs. So this work is the work of Daniel Matute. We talked perhaps a little bit about it yesterday. We confirmed it over and over again, also with newly collected strains from the laboratory. We can also see this phenomenon evolve within 5 to 10 generations. If you put these two species together and you let them live together in cages and hybridize or not hybridize, you see that in 5 to 10 generations, those females will, in Drosophila ecuba, will dump the eggs of the other species and come back into mating condition. So we do have reinforcement. It's just not a kind of reinforcement that we're used to seeing. By the way, those cage experiments also show an increase in sexual isolation. So this is a mystery. Why we see only this form of reinforcement in nature, but we see two forms of reinforcement in the laboratory: sexual discrimination as well as this post-mating phenomenon. Okay. Everybody wants to know about introgression. How much gene flow is there between species? Are there islands of introgression, or are they mirages, as Muhammad has called them? And our work on the introgression of these flies was stimulated by two things. First of all, the arrival of